So it is my pleasure to introduce Katerina Makova. So Katerina is my neighbor. She's at Penn State, which is down the street for us. And she's did her master's degree in Kiev. She did a PhD in Texas. She did a postdoc at the University of Chicago in their Department of Evolutionary Biology, which is one of the best in the country. And she's now at Penn State. And I've been waiting for a long time to hear about her work on sex chromosomes, and I think we'll all find it very interesting. Thank you, Katerina.
Thank you for inviting me. It's a great pleasure for me to be here. Let me see if I can share my slides. I hope so. Okay. Okay. For some reason it's paused. Let me stop share and then I'll reshare again. Yeah. Can you see my slides now? Yes.
See them.
Yeah, you can see them. Excellent. Okay. Yeah, so I'm originally Ukrainian, but I lived in the United States now for almost 30 years. So it gives me really a great pleasure to talk about our work to Ukrainian scientists. And I'm going to talk about the large project, which is highly collaborative. So it's a result of work of many scientists. And it's on the complete sequences and comparative analysis of grade A sex chromosomes. So very recently, in 2022, the complete sequence of the human genome was determined. It's called telomere to telomere, or from one end to another end. I quickly noticed that only the sequence of the human female genome was determined. So the very interesting Y chromosome was still missing. Still, it was a major achievement. It was a real milestone in genomics to be able to read the complete human genome, even though it's a female genome. So about 8% of sequence, all of the sequences in red were added. And it really is a major achievement. So I contacted the lead authors of this paper, asking whether they were working on the sequencing of the Y chromosome. And they were working on this. So they actually invited me to join this telomere to telomere consortium, in which we then sequenced the human Y chromosome. The human Y chromosome is a very difficult chromosome to work with because it is highly repetitive. Nevertheless, using lone sequencing reads, we were able to complete the sequence and also to analyze the human Y chromosome. This is our publication from a couple of years ago. And now it is a single contig of about 63 megabases long. There are 0 gaps. It's a complete sequence. And with this new assembly of the human Y chromosome, we gained 30 megabases and 41 protein coding genes. And we deciphered this whole heterochromatic region, which is full of satellites and repeats. You would say that gaining 41 protein coding genes is not a lot, but for the Y chromosome, it is a lot because it actually is very gene poor. So if you're interested in this work, this is the QR code. You can take a look at it. There is even, I gave an interview for NPR, a National Public Radio program here in the US. You can listen to that as well. So now both human X chromosome and human Y chromosome, the 2 sex chromosomes, are in the T2T state, complete sequence for them was deciphered, telomere to telomere, and they are the pair of our sex chromosomes, yet they are very, very different. The human X has 154 megabases and carries about 800 protein coding genes, while the human Y is 63 megabases long and carries only 100 protein coding genes. So the question is, why are they so different? Why do they differ so much in size, but also in protein coding gene content? And to address this question, we need to revisit the evolution of sex chromosomes in mammals. It all started about 170 million years ago, when a pair of regular autosomes acquired a mutation in one of the genes. This gene is called SOX3, so that this gene became the male determining gene, the male sex determinant gene, or SRY, here is shown in brown. And then this chromosome that carries the SRY gene became the Y chromosome. And it started accumulating genes and mutations beneficial to males and harmful to females. To fix these mutations and these genes, the Y chromosome underwent a series of inversions so that it could not recombine with the X anymore. And it started accumulating deleterious mutations. It also lost a lot of sequence. It also accumulated many repeats. It degraded overall, so that now the X and Y recombine only over a very short region called pseudoautosomal region. Otherwise, they do not recombine with each other. So this explains the loss of genes on the Y and also the difference in size between the X and Y chromosomes. So this is the structure of the human Y chromosome as it was deciphered more than 20 years ago. This is the incomplete sequence, but still the major classes of the human Y chromosome could be determined. So I'm going to be focusing on just three groups of regions. The one group of regions is the pseudoautosomal regions. So they are shown here in green and also the other tip of the Y chromosome. And these are the regions in which the Y still recombines with the X. Then there are these yellow regions. They're called the X degenerate regions. And this are the remnants of the Y chromosome past that it shared with the X chromosome. But then there are also these purple regions. They are the amplicodic regions, and this is where the Y chromosome has multi-copy sequences. So this multi-copy sequences, or the amplicodic regions, they have genes that function in spermatogenesis. There are a total of 9 gene families. They are shown here with their functions. Most all of them are important for spermatogenesis, and if some of these genes are deleted, this can lead to infertility phenotypes in males. So one of the genes, one of my favorite genes because I studied it a lot, it's called DAS, or deleted in azoospermia. If this gene is deleted in humans, this leads to lack of sperm and can lead to complete infertility. The interesting thing about these genes is that they are frequently located in palindromes. And palindromes are these sequences that can be read both forward direction and then they're repeated in the opposite direction. And this leads to gene conversion occurring between the two copies of these sequences. So we actually studied the ampliconic genes on the Y chromosome in a sample of 100 males from around the world. And here on the left, we built the phylogenetic tree from these sequences using single nucleotide polymorphisms. And this is a very nicely resolved phylogenetic tree. But on the right, we built the tree based on the copy number of these genes. And based on the copy number of the genes, there was very little clustering. And almost every man had a unique combination of copy numbers for this 9-gene family. So almost every man is unique in how many copies of these genes he has. We only had 2 individuals who had the identical copy number, and yet they were not phylogenetically related, suggesting that this was due to convergence. So why are sex chromosomes interesting? This is a question. The Y chromosome harbors azoospermia factor regions, deletions of which can cause infertility in men, just as I mentioned the S gene. But also the Y chromosome is used to analyze male dispersal in natural populations, and this is important for endangered species. In contrast, the X carries many genes important for cognition and reproduction. It's called by Jenny Graves, the brains and balls chromosome. And also the X chromosome constitutes 7% of the human genome, but its mutations account for 10% of Mendelian disorders. So it carries disproportionately more mutations that lead to diseases as compared to its size. Another interesting thing about the X chromosome, and this is something relatively new that was discovered by other labs, is that the gene expression imbalance on the X chromosome leads to autoimmune disorders, and autoimmune disorders are more common in females. So despite all this importance of the sex chromosomes, they have been understudied because they are haploid in males. So most genome sequencing projects, they focused on sequencing females because this way, at least you can get a good sequence of the X chromosome. If you sequence a male, then you are not going to get a good sequence of either the X or the Y chromosomes. Also, sex chromosomes have high repetitive content. And also they have palindromes, particularly for the Y, which complicate both sequencing and assembly of sex chromosomes. So one of the observations that we made early on had to do with the gorilla and chimpanzee Y chromosomes. So we published a paper in 2016 in which we deciphered the sequence of the gorilla Y chromosome. It was not a complete assembly back then, but still it was a decent assembly. And our conclusion was that the human Y chromosome resembles gorilla Y more than it resembles the chimpanzee Y chromosome, even though human and chimpanzee shared the most recent common ancestor. And this was evident both in terms of the number of genes shared, also in terms of the number of ampliconic gene families, number of palindromes, the repetitive element content. And the big question was why? Why is this the case? It was a bit of a puzzle, but with just 3 sex chromosomes, or just 3 Y chromosomes from human, chimpanzee, and gorilla, we really could not address the question why this is the case. Could it be that the chimpanzee Y chromosome was evolving really fast? Or maybe it was the human and gorilla Y chromosomes that evolved really slowly? We really couldn't tell. So we needed additional Y chromosome sequences from additional apes to really address this question. So in 2020, we published a paper in which we deciphered additional Y chromosome sequences from Sumatran orangutan, from bonobo. Then we used the available sequences from human Y and from chimpanzee Y, but also the gorilla Y we published previously. And what we found by analyzing all these Y chromosome sequences is that the great ape common ancestor already contained many gene families and multi-copy sequences. And then in the common ancestor of bonobo and chimpanzee, there was this accelerated gene death and accelerated nucleotide substitution rates. So this phylogenetic branch really experienced this unusual evolutionary speed up. And this could be related to the mating patterns of these great apes because they experience a lot of sperm competition. Yet we could not really address the questions that are particularly interesting for the Y chromosome evolution. So better assemblies were needed to study evolution of ampliconic regions, of palindromes, of satellites, of transposable elements, also of gene copies and pseudogenes and structural variants. So all of this was inaccessible to us as of just 5 years ago. So that's exactly the question that we asked, is whether we can decipher the complete sequences of great ape sex chromosomes to study their evolution. And to address this question, I approached the telomere to telomere consortium that was previously working with the human genome, as well as some other scientists. And together we formed the telomere to telomere primates consortium that is currently co-led by myself, Adam Philippi, and Evan Eichler. It includes scientists from 14 different institutions. We are close to 90 people right now. So it's really a very large collaborative effort, and we share all the data with anyone who is interested in using the data. Our data are on GitHub as soon as we generate the data. So it's a very open consortium as well. So let me talk more about the study design. So these are the ape species that we selected for generating the telomere to telomere sex chromosome assemblies. They include bonobo and chimpanzee, which separated from each other about 2.5 million years ago, and separated from the human lineage about 7 million years ago. Gorilla, which separated from the human lineage about 9 million years ago. Also 2 species of orangutans, Bornean and Sumatran orangutan, which separated from each other about 1 million years ago, and separated from the human lineage about 17 million years ago. We also included an outgroup, a lesser ape. It's a siamang, and it separated from the human lineage about 20 million years ago. So these species, they really differ in the level of sperm competition. For chimpanzee and bonobo, it's quite strong. For gorilla orangutans, it's very weak. Siamangs are monogamous, so there is a weak sperm competition, if any. And also there is a different mode of dispersal for this species, either by females or by males, or together by males and females. So it's a very diverse data set, yet it includes almost all extant great ape species. All of them are our closest relatives. So what were the experimental methods that we used? So we worked with cell lines that we acquired from Coriel, San Diego Zoo, and also the some collaborators such as Lucia Carbone. We propagated cell lines here at Penn State, and for a while we became the cell line distributing facility. All the cell lines were carrier typed at the National Human Genome Research Institute at the NIH. And then they were sequenced using mostly long read technology, the PacBio Hi-Fi, the Oxford Nanopore technology, also Hi-C Illumina technology. And the sequencing depth was quite high, so that this really helped the assembly. And for two of the individuals, we had samples from the parents. So this was sequenced with short read Illumina technology. And this again helped the assembly. So this was all in collaboration with our colleagues at NIGRI. And computational methods, we used this software called Verco. And it uses the PacBio reads to generate the deployed assembly graph. And then this graph is further resolved using alignments of Oxford Nanopore Technology Labs. And then to really resolve the haplotype specific nodes, we either used information from parents, parent-specific k-mers, or we used high-C assemblies if we didn't have information from trios. So this is the work from Adam Philippi's lab. So this is how the sex chromosome assemblies look like. These are assembly graphs prior to manual curation. And the X chromosome is in red and the Y chromosome is in blue. So they already, even prior to manual curation, assembled quite nicely. But then we finished their assemblies manually. It required some manual curation. So what we did altogether is we generated 4 new telomere to telomere chromosome assemblies and 8 improved telomere to telomere chromosome assemblies. So what you see here in black are the sequences that we added to the previously existing assemblies. So the Bornean orangutan X chromosome was not available before. So all of the sequences in black, or the new sequence. Simon X was also unavailable. It's also in black. But then even for the X chromosomes that were available previously, we added quite substantial sequencing information. For example, for Gorilla X, we completely sequenced the centromere, and we also sequenced the telomeric regions and subtelomeric regions. But major improvements were certainly for the Y chromosome. So for the Y chromosome, you can see just how much of sequence we added. And many of the previous assemblies were our own assemblies in our lab, so we improved a lot upon them. But the Y chromosome is so difficult to work with that we're very happy with the results here. So the biological results. There is little variation in the size on the X chromosomes, but there is much more variation in size for the Y chromosome. If you compare the largest versus the smallest Y chromosomes, its Siamang Y is 30 megabases, while the Sumatran orangutan Y is 68 megabases. It's a twofold size difference. But overall, it's large variation for the Y and less variation for the X. And this is going to be the common theme of the observations that I'm going to present to you for the rest of my talk. So some other interesting observations is that human and bonobo each acquired independent pseudoautosomal regions, while the first pseudoautosomal region, we call it PAR1, is shared across all apes that we studied. Human and bonobo have additional pseudoautosomal regions that are not homologous to each other. So over very short evolutionary time, pseudoautosomal regions can be acquired. We also collaborated with Jen Gurton at Stowers Institute on studying the ribosomal DNA arrays. This, again, is a very difficult to study region of the genome. And we detected the presence of ribosomal DNA arrays on the Y chromosome in both orangutans, but also in Siamang. And this really highlights the different number of copies of ribosomal DNA between males and females. There is an increased abundance of ribosomal DNA in males of some animals, such as orangutans and Saiman. And these arrays are also known to be active because they are methylated. So this might give some advantage to these animals when their ribosomal RNA is being produced. So we made many, many important observations from this amazing data set. So today I'm only going to focus on 2 observations so that they will be your take-home messages. And the first observation is that there is higher interspecific variability on the Y than on the X. And I'm going to highlight this using a series of different features of sex chromosomes, such as alignability, nucleotide substitution, structural variants, segmental duplications, and repeats. So here, what we did is we aligned the chromosomes in pairwise fashion, and the alignments for chromosome X are on the left, and the alignments for chromosome Y are on the right. So you can see that the X chromosomes align very nicely between species. And human versus chimpanzee, you can align about 97% of the chromosome sequence between them. This is really nice and what one would expect. But if you look at the Y chromosome, it's very different, right? It's very different. So only about 14% of the human chromosome can be aligned by the Simon Y chromosome. And if we compare human with chimpanzee, then human to chimpanzee, it's about only 26% of the chimpanzee chromosome that can be aligned by the human Y chromosome. So only about a quarter of the chromosome that can even be aligned. So this is really the observation that we could not have found in with any other chromosomes that we looked at. So the Y chromosome is really unique, that it has such a low alignability, while the alignability for the X chromosome is very high. Then we built phylogenetic trees based on nucleotide sequence divergence. And the tree for the X chromosome is shown on the left, and the tree for the Y chromosome is shown on the right. And the scale for these trees is the same. You don't have to be a statistician to appreciate that overall branch length is much longer for the Y chromosome than it is for the X chromosome. And if you look at the human chimpanzee divergence, it's about 1% for the X chromosome, which is what you find in textbooks, right? This is the number that is frequently used in textbooks. But human chimpanzee Y chromosome divergence is 2.5%. It is much higher. So to conclude, there is a higher nucleotide sequence divergence on the Y than on the X. There is also more rapid accumulation of structural variants on the Y than on the X. So here we aligned human X chromosome with each of the ape X chromosomes. These are very nice alignments. And here for you to see the alignments of the human Y chromosome to the Y chromosomes of other apes. It's a very, very different pattern, very little of the human Y chromosome can be aligned to the other Y chromosomes. But then we studied the speed of accumulation of structural variants, and these are the 50 base pair to 300 kilobase pair insertions, deletions, inversions, and duplications. And the speed of accumulation is shown for the Y chromosome in blue and for the X chromosome in orange. And you can see that the slope is much higher for the Y chromosome than it is for the X chromosome. So again, there is a more rapid accumulation of structural variants on the Y than on the X. Segmental duplications, these are repetitive more than one KB segments with at least 90% identity. The X chromosome is covered by only about 4 to 7% of its sequence in the segmental duplications, but segmental duplications cover 23 to 56% of the Y chromosomes, much, much higher percentage. Sometimes more than half of the Y chromosome consists of segmental duplications. And here they are shown in blue, the segmental duplications within the same chromosome. So many more segmental duplications on the Y than on the X. And the other thing that I would like to highlight is the repetitive element content on the Y and on the X. Just based on my slides about the evolution of the Y chromosome, it is expected to accumulate repeats. So we knew that it will be repetitive, it will be highly repetitive. But we just didn't know how repetitive it will be. So just to give you a benchmark, about 54% of human autosomal T2T sequences is annotated in repeats. So more than half of our autosomal sequences consist of repeats. We look at X chromosomes, 62 to 66% of them are in repeats. But for the Y chromosome, it's 71 to 85% of them are in repeats. So this is really, really very high percentage. And this is shown on the graph here. So these are the X chromosomes and their repetitive element content. We see an expansion of line elements. And these are the Y chromosomes. They have even higher repetitive element content, and they have expansion of simple and satellite repeats. And when we look at satellites in particular, there is a lot of variation in different types of satellites among different Y chromosomes. So, for example, 1/3 of gorilla Y chromosome consists of satellites, which are particularly overrepresented in ST sats, and 1/3 of Simon Y chromosome consists of satellites as well, but these are different satellites. These are mostly alpha satellites. So your take-home message from this part of my presentation is that the Y chromosome has a remarkably dynamic evolution, especially in contrast to the X chromosome, for which the evolution is much more slow. And this is evident from sequences aligned, nucleotide substitution, structural variants, segmental duplications, repeats, and satellites. So the big question is why? Why does Y chromosome experience such a dynamic evolution? And we have several hypotheses. One is that there is a larger number of replications and more mutations in the male germline, the phenomenon that is called male mutation bias. Another potential explanation is that there is no recombination outside of pseudoautosomal regions. So any mutations that the Y chromosome acquires, they cannot be corrected by the second copy of the Y chromosome because the second copy of the Y chromosome does not exist. Also, the Y chromosome has a small effective population size, which increases the effects of random genetic drift. And there are some other evolutionary processes such as Miller's ratchet and selection that might contribute to highly dynamic evolution on the Y, and I can talk about them if somebody has a question. So I just want to talk more about the male mutation bias, and it has to do with a very different number of cell divisions between males and females. So in females, the number of cell divisions during oogenesis is only 24. And while in males, the reproductive age, let's say of 20, it's 150, and this number increases with males' age. So if mutations originate as errors during the DNA replication, then we expect more mutations originating in males than in females. And the Y chromosome is only present in males, so it will reflect the male-specific mutation rate. So we expect more mutations on the Y, fewer on autosomes, and even fewer on X chromosomes. Okay, so I'm going to move to my second observation, and I'm going to state the obvious. So the Y chromosome is still alive, even though it has this dynamic evolution, highly dynamic evolution, and accumulates mutations at a very high rate. The question is why, and another question is how. And I'm going to talk about the ampliconic regions, palindromes, also genetic redundancy, gene evolution, and selection. So this is the human Y chromosome, which contains this multi-copy ampliconic regions. It has these palindromes, and these palindromes carry genes. So in general, the Y chromosome has a lot of intrachromosomal similarity. So this is the plot of alignment of each of the Y chromosome to itself, and there is a lot of intrachromosomal similarity. It's quite high percentage. So these are 5 Y chromosomes and this additional 2 Y chromosomes. When we tried to build a plot of self-similarity with the X chromosomes, it was almost completely empty. So it was not much to plot. So there is high intrachromosomal similarity on the Y, but not on the X. And the Y has many regions with intrachromosomal similarity. In fact, they constitute about 11 to 56% of the Y overall, depending on species. While on the X, these regions constitute only about 0.15 to 1.1% of the overall X chromosome sequence. So it's a very different pattern between the X and the Y. So why does the Y chromosome have so many sequences with intrachromosomal similarity, the repetitive sequences? And in fact, these sequences on the Y are frequently species-specific or genus-specific, while the sequences on the X are frequently shared across a much larger number of species. So these ampliconic regions must be important for some species-specific adaptations. And here, a postdoc in my group, Carl Paul, did a detailed analysis of palindromes on the X chromosome and on the Y chromosome. And the palindromes are sticking out as the teeth of the comb. They are shown here in black, and the same for the X chromosome. And the Y axis is different for these plots, but the X axis is the same. So what is obvious is that there are many more palindromes on the Y in general than on the X. Also, the palindromes on the Y are longer. They're frequently lineage specific. They are only shared between, let's say, only closely related species, while for the X, they frequently shared all across. So these colored lines represent palindrome sharing. So there are many more palindromes on the Y, and they show a different evolutionary pattern. So another important point about the Y chromosome is that it contains many multi-copy genes. So here I'm just focusing on one of the gene families, TSPY. And this gene family is present in a tandem array on Sumatran orangutan Y, but it is present in palindromes in Siamang Y. So there is this signature of genetic redundancy present for many genes on the Y chromosome, and it is achieved either via tandem array organization or organization into palindromes. And this is the phylogenetic tree for the TSPY gene copies, and different colors show different species. And almost all the clusters are species or genus specific, suggesting that there is a lot of sequence homogenization, most likely due to gene conversion among gene copies. So we looked at the evolution of these ampliconic genes and also some single copy genes. They're also called ancestral genes. And we found a lot of variation in the copy number of Y ampliconic genes. But we also found that both for ampliconic genes and ancestral genes, many genes evolved under purifying selection. So all the genes or gene families that are underlined, they show the signature of purifying selection as based on the analysis of non-synonymous to synonymous rate ratio. So this is the work in collaboration with Sergey Pond, now from Temple University, but originally he's also from Kiev, from Ukraine. So this is very important. So there is still a lot of selection happening on genes for the Y chromosome. So your second take-home message is that the Y chromosome survives, at least in part, because it still carries some genes that are evolving under selection. So it still carries genes that are needed. And how does it survive? Well, many genes are multi-copy and located within palindromes. So even if one of the gene copies is damaged by mutations, then another copy can take over and fulfill the function of this gene family. But also the abundance of these palindromes, of these repetitive sequences in which many of these multi-copy gene families are embedded, it really facilitates intrachromosomal recombination that leads to gene conversion. And this really can mitigate the accumulation of deleterious mutations. So even though the Y chromosome is single copy and it does not recombine with other chromosomes, it evolved to have multi-copy sequences so that it can recombine with itself. And this way it can survive even while being bombarded by so many mutations with the high male mutation rate. So overall, our conclusions is that the Y chromosome is highly variable and has evolved to recombine with itself to survive in the absence of intrachromosomal recombination. We could do this work and this analysis because of advances in sequencing technology, long reads in particular, but also the alignment algorithms and telomere to telomere assemblies. They're a total game changer for sex chromosome studies. And our research is really expected to inform future studies of conservation genetics of non-human apes, all of which are endangered. Our results can be important for evolutionary medicine. What are the features of these great apes and their genomes that we could study in more detail to inform the development of new medical approaches for humans? The nice byproduct of this work is that in addition to completely deciphering the sex chromosomes of telomere to telomere level, we also now have the complete telomere to telomere assemblies of autosomes. This is the work that will be published later this year. And in the future, I would like to do more detailed analysis of ampliconic genes on the X and on the Y. We're working on human and ape pangenomes, on gibbon genomes. We're also studying the sex chromosomes of other vertebrates. So this topic is really interesting, and this telomere to telomere assemblies really open doors for a detailed analysis of sex chromosome that was not possible previously. So if you are interested in this work, this is the publication that came out last year. And this is the QR code for this publication. You can just scan it, or you're welcome to send me an email. I'll send you a PDF. So again, this is a very large collaborative effort. That's not something that myself or my lab could do alone in isolation. I would like to acknowledge our funding by the National Institutes of Health and also by Penn State Eberly College of Science. Yeah, and first of all, and foremost, I would like to acknowledge people from my lab who always inspiring me to do good science and who are asking all these incredible questions. And just recently, we were in Pittsburgh Zoo, where we met all the animals whose genomes we sequenced. So this was really a good experience. So I'm happy to take any questions that you might have. And I will stop sharing.
Thank you, Katerina. Do we have a question from online? Do we have a question from the audience?
Thank you so much. You just touched theme I'm really interested in, so I'm just great. So my question is really simple, because I want to ask what cells, I guess, you take for sequences, because of, if I know right, the Y chromosome is just loses in some somatic cells, because it not make any sense in, I don't know, in skin or other cells. So it's maybe only germ cells produce really full Y chromosome. Yes.
Yeah, I think this is a very important question. So here we used fibroblast cell lines and lymphoblastoid cell lines, depending on species, depending on availability. So all of these are somatic cell lines. And we karyotyped them before we proceeded with sequencing. So we really wanted to make sure that the Y chromosomes are present there, and that overall we didn't have any chromosomal rearrangements due to propagation of cell lines. But you're right. In many cases, the Y chromosome is lost in some somatic cells exhibiting mosaicism. So it's an important consideration.
Thank you. And
thank you very much for the presentation. It's overwhelming. I am a bio, and I'm really concerned now, because you showed us the graph of the lengths of a Y chromosome in different apes and in human, and as I understand, it tends to shorten. In 7 million years, it was shortened by a half. My question is, first question, will this process stop one day, or is it just discontinuous?
I get this question a lot, and I know that it really is an important question. First of all, in humans, there is a lot of variability in the Y chromosome size. So recently, there was a paper that came out from Pili Halast's group, and they sequenced the Y chromosome in 43 different males, almost telomere to telomere. And the size was differed from 40 megabases to 80 megabases, just among different humans. So one of the limitations of the work that I presented today is that we only analyzed one individual per species. There is also a lot of variability among individuals. So something to keep in mind. And the Y chromosome will continue to evolve. And it's difficult to project whether it will be lost in 7 million years or not. But even if it will be, then another pair of autosomes might become the new sex chromosome pair in our species or in other mammalian or primate species. So this is something that was observed in other animals, that some different pairs of autosomes assume the function of sex chromosomes. It's the sex chromosome evolutionary cycle.
Thank you very much. And
Mhm.
question, you said that there are pseudo autosomal regions, and you highlighted that they're present in humans and in bonobo ape monkeys.
Mhm.
Share some behavioral characteristics that are unique for mammals, for all mammals. Do these two facts connect, and are they connected to behavior? Thank you.
Yeah. So all apes that we studied so far, they have homologous first pseudoautosomal region, par1. But then human and bonobo each independently acquired a second one, right? And I would say that this is just an evolutionary accident. I would not maybe overanalyze this observation. And yeah, there are some characteristics that we share with bonobo in terms of behavior, but I don't think that they're linked to the presence of a second pseudoautosomal region in humans and in bonobo.
We have a question from online. Valerie? You're next.
Okay, thank
No,
you.
no, you're next. Valerie?
Can you hear me?
Yes, yes.
Thank you for an amazing lecture. But my question probably was like briefly answered previously, but I still would like to ask it. So nowadays, different conspiracies like flat Earth conspiracy or like young Earth creationist movements are rising, and I do like second chromosome because it harbors like relic of telomere to telomere fusion event in our ancestors. Do we have some similar evidence? Like, can you give something like really simple to present, but still interesting from this study? Like you said about second pseudo autosomal region, maybe something else?
To prove the existence of evolutionary process, or for what purpose?
No, to just like to easily present or even to discover it at your own laptop. Like you can do it with second chromosome, and it's something that easy to show someone who is like adapting in evolution. So maybe like you have more examples that we can do at home in like in discussions with the people that we met and who are like against evolution.
Yeah, so the evolution is so much embedded in me that sometimes I find this difficult. Yeah, but I think that one example that could be used here is this different rate of evolution for bonobo and chimpanzee for the Y chromosome as compared to other Y chromosomes, right? So the bonobo and chimpanzee really have this evolutionary rate increase, and this is linked to their behavior. So this is something that even the general audience can relate to, that we can see this signature of behavior and adaptation to behavior when we look at DNA sequence evolution.
Oh, thank you.
Yeah.
We have another question from you.
Okay, so thank you so much for the opportunity to speak. So basically, I remember that you were talking about death-related mutations and that it basically influences fertility. So have you studied the biological processes that like a consequence of this? And if no, do you have some suggestions why we have infertility because of that? I mean, like death mutation. So yeah.
Yes. So this is not directly our work. I mean, the clinical part, we just look at the evolution and sequence evolution. But this is an observation that comes from the in vitro fertilization clinics and other clinics. And these observations are very strong and show that particular regions of the Y chromosome are deleted in individuals with infertility. So how this can be studied? This is studied in patients, right? In many cases, this, unless there is some intervention, unless there is some assistance for such individuals, these combinations of Y chromosomes, they will not go into next generation, right? So you actually will not observe individuals in the wild, right, in great apes who have lost a complete gene family that is important for fertility. So my overall suggestion would be to collaborate with clinicians, right, to really investigate the genes that are important for male fertility that are located on the Y chromosome.
Thank you so much for your answer.
We have another question from online, Tatiana.
Thank you very much for your presentation. It's very interesting. And actually, my question, if I understand correctly, if there is the mechanism of restoring these numerous gene copies by gene conversion. And actually, is it known something? How is it the original copy is determined? How the template copy selected for this process? Thank you.
Yeah, Tatiana, I think that this is a very interesting question and a very important one. So my understanding that this is this happens by trial and error, right? So it occurs randomly. It can be that the damaged copy sometimes becomes a template, right? And the functional copy is corrected, right? But then it's not helping anything. Or in some cases, in some lucky cases, the functional copy is used as a template, right? And then the non-functional copy is corrected. But then the selection is deciding what will be kept for the next generation. Yeah. So it's first, it's gene conversion, right? Happening randomly, right? Without reference to what is functional, what is not. But then the next step, selection, should decide what will be kept in the population.
We have another question from the audience.
Yeah.
Thank you.
Thank you very much.
Beginning of your presentation, you said that higher levels of sperm competition inside of the populations can be a possible reason of those drastic reconstructions and changes in the Y chromosome. So my question is why and how? Because as I understand, high levels of sperm competition can also affect, I think, other chromosomes and other proteins that may be involved in the reproductive behaviors. Thank you.
Yes, absolutely. You are absolutely correct. And this is a hypothesis that we have. We do not have direct proof of this hypothesis. We looked at patterns of selection for many of reproductive genes. We particularly focused on the sex chromosomes, but we definitely should look also at genes of the other chromosomes. And so far, we have not detected signatures of lineage-specific selection for such genes. However, we know that we lack power. We lack statistical power. All of these genes and sequences are very similar to each other. We're talking about 2.5% divergence. So we simply lack power to do this analysis. And we are turning to analysis of multiple individuals per species, where we should have more statistical power. So I think I'm just not ready to answer your question right now. We should do more research and more work to investigate it.
Okay, thank you.
Is Alexander Yushuk online yet, our next speaker, or do we have time for another question? Alexander, are you with us? Do we have another question in the audience?
Yeah. Thank you for your presentation. And you mentioned that you're studying also in other vertebrates than apes. And my question is it common in species where sex is determined by chromosome, that one of the sex determinating chromosome is more dynamic, going through more dynamic evolution? Like, for example, in the birds.
Yes, yes. So this is something that we're still investigating, right? So in birds, there is the ZW system. Yes, so W is actually female specific sex chromosome. Yeah. So the evolutionary dynamics will be different, but still W is the chromosome that is present in the heterogametic sex. It also accumulates repeats, so and loses genes. This is definitely something that we observe. But additional studies of sex chromosome evolution in other species is something that needs to be done. Obviously, primates have been investigated much more than other animals.
And another one question is about, you said that you have open files of the sequences, and I want to ask if all proteins there are annotated. Yes,
Yes,
thank
yes. All the genes have been annotated. And for the Y chromosome, it was a very tedious and very difficult process. We did a lot of manual annotation on top of the automated annotation that was provided by NCBI. All of this is available with our paper, and you also are welcome to contact us if you have any questions.
you. Your great work. Thank you.
Thank you. Mhm.
Let Katerina, thank you for a wonderful talk. Let's thank
Thank
Katerina.
you. Yeah, feel free to get in touch with me if you guys have any questions by email. Mhm.
Is our next speaker with us?
Yes. Hello, everyone. Do you hear me?
Can hear you. So
Okay.
are you set up?
Yeah. I'm ready to start my talk.
Would you like me to introduce you?
Of course, if you want.
Our next speaker is Alexander Yashuk from Ivano-Frankivsk University. So he got his master's degree at Ivano-Frankivsk. He then went
Yeah.
to PhD in Tübingen and did a postdoc in Italy. And now he's back in Ivano-Frankivsk, where he's working on finding new antibiotics that can be used against multi-drug resistant bacteria, which is a very, very important question. And I'm looking forward to hearing what you have to say.
Thank you for the introduction. Anyhow, I'm from Lviv. Not sure why you have info about Ivano-Frankivsk, but anyway, I will start my talk. And do you see the slides already?
Not yet.
Okay. Oh. Yeah, I have some blinking screen. Do you see these slides now or no? Okay, once again. Okay, and now?
Not yet. We see that
No,
you're
I will. Okay, I will probably be sharing just the screen because I always have these problems, probably because of my PC or whatsoever. Okay, so now you should see at least, I hope, something. Uh, so do you see my screen?
Yep.
No.
your screen.
Mm.
I think once you went into presentation mode, it disappeared again.
Yeah, just 1 second. There is something seems to be wrong. Okay. And now? Okay. So can you see my screen now?
Yes.
Very well. And now does it move if I change the slides?
Yes.
Okay, okay. Sorry for this delay. And today we are going to talk about some insights into the evolution of antibiotic production and antibiotic resistance in soil bacteria. And so let's imagine the world before antibiotics, and we can ask, was it really a good world? And probably it was, but not really a long living expectancy was in that world. And if we recall the pre-antibiotic world, the first thing that comes to your mind is the Black Death. But even more problematic bacterial infections were, except of, for example, tuberculosis, were venereal disease. And this is something that we tend not to talk about, but they were taking an extraordinary toll and decreasing the life quality of millions of people worldwide. And venereal diseases were one of the main, let's say, drivers of the search for new antibiotics, because we know that first semi-synthetic sulfonylamides were actually designed to treat some venereal diseases. And however, the real golden era of antibiotics started when the natural compounds were discovered. And if I have the feedback from the audience, I would like to ask you if you know who is this very venerable old gentleman in glasses. So maybe somebody can tell me who is this guy. Okay, probably I don't have feedback with the audience, but this is Sir Alexander Fleming, the discoverer of penicillin. And right near to him, you can see another important scientist in the field of antibiotics, and this is Salman Waxman, which we are going to talk slightly later. And indeed, the first antibiotic that was mass-produced was penicillin. But after the discovery of penicillin and gramicidin, which is a Soviet discovery, we have a significant explosion of the novel antibiotics that unfortunately decreased in modern times, because less and less antibiotics are reaching clinics maybe due to some problems with the novelty. Some will say that not all modern compounds can pass all the strict preclinical and clinical trials, all possible requirements, because, for example, if you check some of the antibiotics here, and you will see glycopeptide antibiotic named vancomycin. Just after vancomycin, you would find another similar antibiotic, which is called ristomycin. And ristomycin, in fact, was approved for clinical use in United States and even used for several almost a complete decade, but this antibiotic was causing significant platelet coagulation. Antibiotic after the results of significantly more comprehensive clinical and preclinical trials will appear. But at that time, many antibiotics were allowed for the application as is, because you know, you didn't have that much antibiotics at the time. And so the appearance of various antibiotics actually, if you can see on the graph, actually significantly decreased the total death toll caused by infectious diseases, while the non-infectious diseases remained almost the same. Overall, it is estimated that the application of antibiotics allowed to save more than 200 million lives. If we start to count from the appearance of penicillin and up till now. So it is hard to overestimate the importance of antibiotics in everyday life. And just going for some additional trivia, let's talk in more detail about penicillin. So if you know that penicillin that was discovered by Alexander Fleming comes from a slime, a mold, not slime, but just a mold that is called Penicillium chrysogenum, or it was earlier called Penicillium notatum. And this is the structure of penicillin. And you can see so-called beta-lactam ring here. All penicillin-like antibiotics which are carrying this beta-lactam ring, which is, let's say, the warhead of this antibiotic, are called beta-lactams. And unfortunately, although the early history of penicillin was quite interesting, because British were unable to scale up the production of penicillin, and there were even anecdotal cases when penicillin was recovered from the urine of the treated person, purified once again, and used to treat another person. But now no large-scale production was possible in UK, so only in cooperation with USA, it was possible to scale up the production of penicillin, to create reliable overproducer strains, and to design, let's say, close to modern ways to mutagenize the producer strains in order to create overproducers. And the real boom of the application of penicillin was the end of the World War II. It is around 1944, because at that time it was already possible to scale up the production, and penicillin saved plenty of millions, probably, of lives of soldiers from the Western side. And the same could be told about gramicidin, which was used by Soviets from the Eastern side. But starting with the very application of penicillin, the signs of the resistance was observed. And first, these were the penicillin-resistant staphylococci. As you can see here, you have Staphylococcus aureus, which is gaining the resistance to penicillin by creating resistant cell wall biosynthetic enzymes, because penicillin inhibits cell wall biosynthesis. Later on, some other bacteria were showing different ways of being resistant to penicillin. For example, Neisseria gonorrhea, which either produced beta-lactamases, enzymes that digested beta-lactam warhead, making the antibiotic inactive, or produced drug efflux pumps, which allowed to pump out the compound, making the cell resistant. And in combination, all these resistant mechanisms led to penicillin to be completely obsolete by 80s. So novel beta-lactams and natural or semi-synthetic beta-lactams came to change the situation. But overall, today, penicillin, this good old penicillin, is just an historical example of how an antibiotic, which was a new hope for the humanity, could become obsolete because of the resistance that bacteria evolve, or maybe they already have this resistance, and they can live in presence of this antibiotic. But we were talking about that other guy with mustache, and now let's talk a bit more about him. So this was Salman Waxman, who was a microbiologist and biochemist of Ukrainian origin. And this person actually revealed that the real cornucopia of antibiotics are not molds, but soil bacteria that belong to group Actinobacteria or Actinomycetes, which are now taxonomically properly are called phylum Actinomycetota. And these bacteria come in various shapes and colors, and they are actually able to produce various antibiotics, which, for example, here we have another example. This is the cell wall biosynthesis, and we can see the beta-lactams that inhibit transpeptidases, one of the crucial steps of murine biosynthesis of bacterial cell wall. But all other antibiotics that you have here, or at least almost all, they are of actinobacterial origin. So today we know much more about actinobacterial antibiotics than Salman Waxman, and we know how they are produced. And again, another trivia, we need to understand that all antibiotics that are produced by actinobacteria, they have their corresponding biosynthetic machinery encoded in large assemblages of genes, which are called biosynthetic gene clusters. Here, for example, you have one of such clusters. As you can see, there is 46 genes, and the total length of this biosynthetic gene cluster is almost 80 kilobase pairs. And here you have all possible genes coding for all possible enzymes, which by turn either produce the precursors of this antibiotic or produce the biosynthetic antibiotic core, or make this core to be modified to achieve a certain chemical structure. And these biosynthetic gene cluster, they obey the rule of, as I say, co-co-co. So these genes are co-localized, co-regulated, and co-expressed. And importantly, for example, here you can see in green, these biosynthetic gene clusters often carry antibiotic resistant genes. So if bacteria produces antibiotic, it needs to defend herself against its own product and avoid suicide during the production. And as a result, biosynthetic gene clusters could be one of the sources of antibiotic resistant genes. And it happens that in our lab, we are investigating one class of cell wall biosynthesis inhibitors, which are called glycopeptide antibiotics. Here you have vancomycin. And why these glycopeptides are important? Because they are in the highest priority group of the critically important antimicrobials for human medicine, according to World Health Organization, because these antibiotics are extremely effective, meaning that they have very low mix against pathogens. And also the resistance to these antibiotics is not so widespread at the moment, but it is spreading, and therefore it is very important to realize how it is spreading, why it is spreading, and how does it work. And so you can see that glycopeptides are divided into 5 main classes according to chemical structure, and we have type 1,4 glycopeptides, which are actually binding the cell wall biosynthesis, and recently discovered, okay, it was discovered some years ago, but the mechanism of action for glycopeptide of type 5 was discovered recently, and it was found that they inhibit some other class of cell wall biosynthesis enzymes, which are involved in the turnover of cell wall, meaning called autolysins. And so these type 5 are not interesting because they are not used in clinics, but among type 1s, vancomycin and teicoplanin are antibiotics used in clinics against, including also beta-lactam resistant staphylococci. And also there are some 3 semi-synthetic glycopeptides which are used in clinic as well. And there was a certain consensus about the resistance to these glycopeptide antibiotics, and we know these mechanisms very well. So basically, once again, glycopeptides are binding lipid 2, the diala-diala terminus of lipid 2, which, when bound with glycopeptide, is enabled to enter transpeptidation and transglycosylation reactions. And as a result, such cell wall is unstable and leads to the lysis of the cell. And there are 2 mechanisms of glycopeptide resistance. This is one HOX-mediated glycopeptide resistance and one Y-mediated glycopeptide resistance. So basically, if we talk about one HOX, 3 enzymes, which is 1, H, A, and X, which are encoded in a so-called 1-HOX operon, are coding the remodeling of cell wall, where the lipid 2 terminal diala is changed to DILAC. And as you can see, diala forms 5 hydrogen bonds with the core of glycopeptide. But if we take diala away and if we introduce DILAC, we lose 1 amino group, and at the end we lose 1 hydrogen bond, and such a complex is unstable. And this diala-DILAC cell wall is resistant to glycopeptide antibiotic. The other mechanism is 1Y-mediated resistance. So here, a single enzyme, which is diala-diala carboxypeptidase, cleaves the terminal diala. And at the end, we have the lipid 2 without 1 terminal diala, which still somehow can enter transpeptidation reactions, although such cell wall would not be as stable as the wild type 1. But as you can see, if we take away diala, we lose almost all hydrogen bonds. And as the result, this would also be resistant to glycopeptide. Another important thing is that either 1-HAX or 1Y mediated cell wall remodeling is a very stressful process for the cell, and it uses many resources. And there is another group of genes which are called 1RS, which is a sensor histidine kinase and the response regulator. And this kinase is able to sense the presence of glycopeptide outside the cell. Then it phosphorylates the response regulator, which by turn starts the expression of a Y1Y and 1-HAX, meaning that if there is no glycopeptide, you have GPA-sensitive cell wall. But if at least traces of glycopeptides appear in the environment, you have the start of the expression of 1Y or 1-HAX, and at the end, the cell wall, the resistant cell wall phenotype. And so, having this background, there was a certain, let's say, consensus about the glycopeptide resistance. So first of all, it was believed that GPA-resistant genes are very rare in actinobacteria and beyond, and they are limited to glycopeptide producers, meaning that really why some other bacteria should have glycopeptide-resistant genes if they maybe never meet the antibiotic in the environment, but the producer should have these genes to avoid suicide during the antibiotic production. The second absolutely obvious consensual point was that these GPA-resistant genes are probably involved only in resistance. And the other thing is that since we observe these glycopeptide-resistant genes in pathogens, one hypothesis believed that these genes originated in glycopeptide producers and then migrated to non-producing bacteria, low GC, soil gram-positives, and pathogens. And the other hypothesis was that these resistant genes emerge independently in glycopeptide producers and in OGC, soil gram-positives, and then as a result, they were transduced to pathogens. And all these facts come from very limited genomic data that was available at the time. And some years ago, it was the COVID period and no work in the lab was available. So having thousands of genomes of actinobacteria, I've set up to analyze all of them and to realize if these points were true or maybe they need some revision. And so at the end, after I have analyzed more than 7,000 genomes of various actinobacteria, I have found out that glycopeptide-resistant genes are quite common. So at least 30% of actinobacterial genomes carried one gene. Contrary, only half of the percent of actinobacterial genomes carried glycopeptide biosynthetic gene clusters, meaning that probably the idea that all glycopeptide-resistant genes lie within biosynthetic gene clusters is not true. And the majority of glycopeptide-resistant genes come from non-producing actinobacteria. And, for example, we can illustrate this with two orders, which is order Pseudonocardiaceae and order Streptomycetaceae. So in case of the order Pseudonocardiaceae, which is a very well-known order that gave us tens of glycopeptide producers, we can see that there are organisms that carry biosynthetic gene clusters for glycopeptides, as you can see in lilac color, and they also carry glycopeptide resistant genes co-localized with the cluster, but also somewhere else on the chromosome. But you can have as well bacteria that do not carry glycopeptide biosynthetic gene clusters, but they carry glycopeptide resistant genes. And some of them, for example, as you can see here, Scibdellosporangium phytohabitans, doesn't have the biosynthetic gene cluster, but it has plenty of glycopeptide resistant genes, much more than these organisms that actually carry clusters. And contrary, if you have this Streptomycetacea order, which is not a source for glycopeptide antibiotics, only for type 5 ones, which, as you know, are not able to inhibit cell wall through lipid 2 inhibition, and therefore they do not require one genes for the resistance. So all these Streptomycetes, they carry different versions and different combinations of glycopeptide resistant genes, but they do not carry any glycopeptide biosynthetic gene clusters for type 1, 2, 3, 4 antibiotics. So we don't see correlation between the presence of glycopeptide biosynthetic gene clusters and glycopeptide resistant genes. Moreover, if you take another order, which is Pseudosporangiaceae, and you have here genus Nonomuria, which are again well-known producers of glycopeptide antibiotics, you can see that almost all Streptosporangiaceae carry one Y gene and one RS, but the ones that carry glycopeptide biosynthetic gene clusters, there is this version from the chromosome that is present in all other members of the order, but there is also another set of 1Y and 1RS genes which are present in glycopeptide biosynthetic gene clusters. And there are also some nonomurias that have this set on the chromosome. They have biosynthetic gene cluster, but they do not have the 1Y gene inside. And again, here, this is another example when the cluster is co-localized with this version, which is actually the chromosome version. And so, also by analyzing the phylogeny of all these, we came to a speculative hypothesis that actually here we can observe a very interesting situation when we have, if we take this clade, we have the last common ancestor of all these strains that carries 1Y, 1RS genes on the chromosome. And we have this protocluster, which is the ancestral to all these 3 glycopeptide biosynthetic gene clusters. And we see that if this protocluster originates from some other nonomuria we don't know, at some point it recruits the copy of the 1Y from the chromosome of this unknown nonomuria. And then this cluster is transferred by horizontal gene transfer events the other nonomurias already carrying this 1Y gene inside that carry this original copy on the chromosome. So here we see that it looks like that maybe not biosynthetic gene cluster is decisive for the presence of genes, but instead the presence of resistant genes are maybe an adaptation for the biosynthetic gene cluster to appear in certain bacteria, because this bacteria is already having the capabilities to be resistant to this antibiotic. And furthermore, we have seen this on various reconstructions, but here again, this is the phylogeny of these 1Y carboxypeptidases. And you can see that some of these organisms are highlighted in red, and these highlighted in red are carrying glycopeptide biosynthetic gene clusters. And it was believed that probably all 1Ys and also 1Hox other genes that are present in glycopeptide biosynthetic gene clusters are monophyletic. But you can see that they are lying very far away from each other on the tree, and they are diluted with some other non-cluster situated 1Y, non-cluster encoded 1Y proteins, meaning that this probably means that these biosynthetic gene clusters, they acquired the resistant genes independently many times, meaning that perhaps when the cluster comes to the organism by horizontal gene transfer event, it first relies on the already present resistant genes, and then it might recruit some of these genes inside its own frame of the cluster to correlate better these three co-co-cos, right? Co-expression, co-regulation due to co-localization. And another thing that comes in mind when you see this tree, and also the same for trees for 1Hox, is that there are much more resistant genes than biosynthetic gene clusters. And this comes quite unusual, but we believe now and that probably, for example, these 1Y resistant genes and some other, they are not resistant at the first place. So these are probably some carboxypeptidases which are used by bacteria to remodel cell wall somehow, for example, due to some environmental triggers. But as a byproduct, they can offer resistance to glycopeptide antibiotics, and therefore they could be recruited by biosynthetic gene clusters. And another thing that was quite interesting, as you can see here, when we screened all these 7,000 genomes, we have found plenty of new biosynthetic gene clusters. And as a byproduct, we have analyzed some of them. First, in silico, we have prioritized them, and then we obtained the production streams. We were able to find the production conditions, purify and chemically characterize them. And at this point, this, let's say, large work about bioinformatics analysis of resistant genes yielded us two novel glycopeptides, which is A5926 and so-called kineomycin. Kineomycin is quite interesting because it doesn't fit into this 5-type classification because it lies somewhere between type 1 and type 2. And also the one from Amyclotopsis bartonia, which is currently in the analysis. We already described this biosynthetic gene cluster, and we have found out that there is certain cause for this biosynthetic pathway to be silent, and now we are currently trying to activate this biosynthetic cluster. But moving away from these byproducts, the next obvious step was, okay, if we have the analysis within actinobacteria, and there are plenty of biosynthetic gene clusters, glycopeptide resistant genes, we need to go deeper and analyze all other available genomes of other bacterial phyla to understand whether they carry these resistant genes, and to estimate the relatedness between the ones from actinobacteria and the ones that all other bacteria carry. And we have also found some interesting new knowledge in this, because first of all, we have found that there are, okay, knew that Clostridia and Bacilli carried glycopeptide-resistant genes because many of them are clinical pathogens, and we knew that pathogens have glycopeptide-resistant genes. But we have found out that some other soil bacteria of unknown clinical significance, which is phyla or classes Anaerolinia, Ctidonobacteria, and Erysipelotrichia, they also carry glycopeptide-resistant genes. And in case of Erysipelotrichia, which are gram-negatives, it's no obvious explanation why they need these genes, because they are already resistant to glycopeptides, being gram-negatives. Moreover, we have found that at least in some Ctidonobacteria, these resistant genes are placed at the regions of the chromosome, which are extremely enriched with insertion sequence-like elements. And this probably explains why do they have these genes. And the other interesting thing about these Ctidonobacteria is that one genes present here actually are evolving. And for example, you can see that neither has 1X, 1X, which is dipeptidase cleaving diala before DLAC is installed. But they have some other unfamiliar peptidase instead. So it is unclear whether these genes can allow Ctidonobacteria to be glycopeptide resistant, because Ctidonobacteria are very under-investigated and hard to work with a class of bacteria. But it seems like they are somehow repurposing these genes to their own needs by changing the composition of these genes. And at the end, we also wanted to understand how these, as I already told, newfound genes are related to the ones from actinobacteria. And so we built the quite global phylogeny of all of them, trying also to correlate these phylogenetic relationships with the genomic context of the genes. And there were quite interesting findings, because first of all, you have here this core actinobacteria group. And we have found out that almost all other groups of gene, which are found in various classes, including Bacilli and Clostridia and so on, are actually very closely related to these core actinobacteria, not allowing to tell that they've evolved separately. No, they all are sister clades to the ones from core actinobacteria, and they were likely mobilized in ancient times from the pool of core actinobacteria and delivered to various groups. But very interesting was that these Ctidonobacteria, genes that we were talking before, they are actually sitting deep in the core actinobacteria gene clade, meaning that if all the other horizontal gene transfer events were quite ancient, these horizontal gene transfer events were very early and probably happened not so long ago, meaning that if it happens now, it proves that it happened before. And moreover, by correlating all these phylogenetic reconstructions with the genomic context, we were actually able to understand some vectors that took these genes from core actinobacteria to all other organisms. At the end, we have built this speculative scheme showing that if we consider the phylum Actinobacteria as an initial main reservoir of one genes, we can actually trace how these genes passed to all other groups and which either transposon or some plasmid or phage, which vectors were taking them. And funny enough, but you can see that, for example, these bacilli, they have received the one genes from Actinobacteria, but they transferred their own one genes to some other bacteria. They have transferred these one genes from one bacilli to another. And at the end, they have even transferred one genes to some groups of Actinobacteria back again. So you can see that this is something quite active, quite huge, and probably not that anthropocentrically driven as we believed before. So to have like some preliminary conclusion on this, and then we will talk shortly about some experimental things. We can say that GPA-resistant genes are widespread in GPA non-producing Actinobacteria, and that these Actinobacteria were the main source of GPA-resistant genes for other bacteria, including low GC gram-positives and the pathogens. But the huge abundance of GPA-resistant genes in Actinobacteria makes us think that probably they have some other physiological roles, which at this point remain unknown. Also, GPA-resistant genes did not originate within biosynthetic gene clusters, but were recruited to them. And they were transferred to non-Actinobacteria by numerous independent horizontal gene transfer events mediated by various vectors. And it seems like GPA-resistant genes are giving some benefits to the acquiring organisms, which are not GPA-resistant, like in case of gram-negative erysipelotrichia. And again, as there are no obvious interaction between some of these rare soil bacteria with humans, we can say that some these forces that are driving the spread of one genes are not so anthropocentric as we believed before. And at the end, we can say that GPA-resistant genes, which probably have other physiological roles except of the resistance, are very important components of actinobacterial pangenome and soil mobilome. And they existed before the discovery and medical application of GPAs, and they transferred, and probably they will do it even when humanity will disappear and we will not use GPAs anymore. And so all this was the comparative genomics part, but as a practical biologist, it was interesting to investigate certain aspects of how these genes work in producing actinobacteria. And we have focused on teicoplanin, which is produced by Actinoplanus teichomyceticus. And teicoplanin producer had the TEI biosynthetic gene cluster, which carries one HAX and one RS homologs, which is TEI75 and TEI24. And by studying them experimentally, we indeed proved that they are operons, that they express throughout the life cycle, and that they are not regulated by pathway-specific regulation. These are regulators that regulate the expression of synthetic genes, meaning that, okay, they are an exception to this co-co-co rule. But analyzing this more, we have found out that although, as I already told you, the consensus is that glycopeptide resistance appears only in presence of glycopeptides. In case of Actinoplanus tecumsehticus, these genes are actually expressed constantly due to high activity of the corresponding promoters. So as you can see, for example, here you have 36 hours of incubation. Glycopeptide is not even starting to produce, but glycopeptide resistant genes are already active. And it was interesting to find the reason for this. And we have compared these histidine kinase T3 and response regulator T2 to the counterparts of model Streptomyces silicular, which is not the producer of glycopeptide, but it has very tightly regulated glycopeptide resistant system that activates in response to vancomycin. And when T2 and 1R were almost identical, there were quite a lot of variations in T3. And these variations were actually happening in including the ATFase domain, and the domain that is crucial for the phosphorylation of the 1R by 1S. And we have hypothesized that maybe these mutations lead to the fact that T3 is not inducible anymore. It is unable to sense glycopeptide, and it constantly phosphorylates T2, and explaining this constant resistance. And at the end, we have found out that indeed, if you express T3 in Streptomyces silicular, which can crosstalk with 1R, obviously, due to very high similarity, this recombinant strain becomes constantly resistant to all studied glycopeptides, meaning that most probably it is a constant kinase. After some gene engineering and creating of some assay systems, we were able to prove that ACT3 actually doesn't require ligand for kinase activity. So here, for example, you see that there are discs with antibiotics and you have collar around them. This is the histidine kinase of Streptomyces silicular. It activates the expression of one genes only in presence of antibiotics. But here you have the whole square green with no regard to the presence of these discs with antibiotic. This is the histidine kinase T3. So it doesn't require any ligand. It simply makes the strain resistant. And again, we again know that we can predict this sensor loop. You can see you have here the two transmembrane domains and this sensory loop, cytoplasmic sensory loop. And this loop in T3 is very different from the one in 1S, implying that it should react on some other antibiotic, presumably teicoplanin. But how can you study the sensitivity of this loop if you have the constant kinase activity? So what we did is we have exchanged the sensor domain of 1S from Streptomyces silicular, which has the inducible kinase activity and known ligand, with the sensor domain from T3. And in this way, we have received the inducible kinase, which is able to react to some kind of ligand. And we believe that this ligand would be teicoplanin. And unfortunately, it was not. We have found out that this sensor histidine kinase, or the sensory domain taken from teicoplanin producer, is unable to react on teicoplanin, but it reacts on A4926, a related glycopeptide. So what is the difference between A4926 and teicoplanin? So basically, as you can see, is the absence of one sugar moiety at amino acid 6 of A4926. Here in teicoplanin, you have N-acetylglucosamine, and here you don't have anything else. So again, as I told you, we can reconstruct the activity of various glycosyl transferases in the biosynthetic gene cluster by phylogenetic analysis. And since probably the cause is the presence or absence of the sugar, we decided to reconstruct the phylogeny of all glycosyl transferases in known glycopeptide production biosynthetic gene clusters. And what we have found out is that we know that T10 glycosyl transferase installs N-acetylglucosamine here. And we know that T1 does it here. But what became obvious from the phylogenetic reconstruction is that T1 is actually a paralog of T10. And most probably in the ancestor cluster, T10 duplicated and diverged. And T1 retained the substrate activity, meaning that, the donor activity, meaning that it will attach N-acetylglucosamine, but it changed the substrate specificity and started to work on AA6 instead of AA4. So now you realize that T10 and T1 is a very recent evolutionary divergence. And the ancestral antibiotic was most probably desglucnacteicoplanin. And desglucnacteicoplanin looks pretty much the same as A4926. So an obvious evolutionary explanation is that protocluster, the ancestral cluster of teicoplanin, had T1 and T10 ancestor, which catalyzed the addition of N-glucosamine at A4. But then you had the gene duplication divergence event, and you now have T1 and T10, which catalyzes the addition of N-acetylglucosamine at two different sites. But how is it possible when our histidine kinase is not able to sense teicoplanin, but senses A4926 only? So here you can see that most probably, first, the mutations in the sensor histidine kinase occurred, giving the strain the constant glycopeptide resistance phenotype. And this gave, again, the space for the biosynthetic gene cluster to evolve and to exhibit this gene duplication divergence event. So if this duplication divergence event would have happened in the strain carrying inducible histidine kinase, this strain would probably never survive this, meaning that his histidine kinase would not be able to react to novel product, meaning that this mutation would be lethal. But a pre-adaptation that happened in T3 most likely gave the cluster the space to evolve. And which I believe is very interesting because it shows that not only biosynthetic gene clusters shape the glycopeptide resistant genes and resistant genes in general, but the resistant genes can shape the biosynthetic gene clusters, and as a result, they can shape the antibiotic structure and production. So at the end, we can draw three very simple conclusions, and we need to understand that resistant genes are widespread, and they existed before the humanity started to use antibiotics, and they will be used even if humanity will stop using antibiotics. And these resistant genes probably serve some other purposes except of the antibiotic resistance that we do not understand. Also, considering this large amount of in general, resistant genes, which are essential part of bacterial mobilome, we can understand that we will never be able to stop the spread of antibiotic resistance, but we can reasonably slow down it. And finally, we see that not only resistant genes are shaped by antibiotics, but the antibiotics could also be shaped by resistant genes, vice versa. And as I see, it's 7, and I'm ending my talk. I would like to thank my students and PhD students who took part in this work at different steps, and I open for your questions.
Thank you. That was really a beautiful story. Thank you. I see. Do we have time for a couple of questions? This is our last. Can I keep going? I see at least 3 questions in the audience here in the room.
Who was the first?
I don't hear you.
1 minute. Good evening. Thank you so much. And my question will be that we have a worldwide problem of antibiotic resistance. However, we keep using antibiotics significantly more often than bacteriophage therapy to treat the infection. So why antibiotics are better than bacteriophages for killing clinical pathogens?
Thank you very much for the question. I should disclaim that although I'm studying antibiotics, I'm quite a fan of phage therapy against bacteria. And I indeed believe that it could be very promising. Why it is not so widely used as it should be, probably? I think that this is the question of the certain fear of viruses. You know, we understand that there is a huge bacteriophage biosphere surrounding us, and we meet phages every day, and we have, I don't know, trillions of them inside our gut. But we are still a little bit frightened to use them as a therapeutic agent, because maybe they will mutate, maybe they will start killing us, and so on. But overall, I believe that the phage therapy it will probably rival the antibiotic therapy in some 20 to 50 years to come.
Do we have another question in the audience?
I have one question. Thank you for your presentation. And when you're talking about the practical application and some stuff called, when you test the antibiotic and resistance, I wonder, have you ever tested just you can knock out that gene producing, I understand, resistance and to see what happened with just metabolism and will vitality of culture in general, with and compare it with control, and maybe you see some results or not, and what can it mean?
Okay, thanks. So we are a little bit biased here, because if we knock out the resistant genes, we are studying the changes in resistance. And now we believe that probably we need to study something beyond, as you say, to identify some other effects that resistant genes may have on the physiology of the strain. For example, currently we are trying to investigate experimentally some of the, let's say, resistance-like genes and some very specific groups of bacteria to understand whether the knockouts of these genes will influence something except resistance. So overall, it's quite a bright idea because we were unable to come to this for decades, and we were studying only resistance in knockout resistance gene strains, but now we are moving away from this, I hope. Yeah, I hope that I understood you correctly and answered your question.
More question in the audience?
Okay, so first of all, you have mentioned that one please. So you have decided to change the receptor for CT3. So how do you come up with this decision to change the receptor?
Once again, I missed the final. How
So
did I?
you have been talking on the slide
Yeah.
in properties of C3 as homologies, so that you basically have decided to change the receptor for T3. So how do you come up with this idea?
Well, I don't know, probably because these two, okay, so these two proteins, they are absolutely collinear, meaning that there are no indels if you, and having two collinear proteins which with high degree of an acid sequence identity, you believe that if you exchange one part to another, it will probably not that likely impact the folding and might work. And we tried to do this and it worked. Yeah, so probably that's how we came to this idea.
Thank you so much.
Thank you.
The audience? I have a question. So you mentioned at the beginning of your talk that actinobacteria are the cornucopia of antibiotic resistance or of antibiotic candidates. And I'm curious, what is it about the biology of the Firmicutes that makes them, they're similar in that they're gram positive and they're similar in that they're soil dwelling, but if they're not a cornucopia, what is it about them that's different?
I missed who is not the cornucopia?
Firmicutes, the low GC.
Gram negatives, you mean? No, you mean bacilli. Okay.
Clostridia.
Or Clostridia. All right. So if you take the average size of actinobacterial genome, you will probably be somewhere in range from 8 to 12 mega base pairs, which if you take Clostridia or Bacilli, it would be around 5. So the average genome of actinomycete is 2 to 3 times larger than the genome of low GC soil gram positives. Why is such a big difference? Because genomes of actinomycetes, they carry dozens of biosynthetic gene clusters for various antibiotics. Some of them are active, some of them are silent, and some of them are cryptic, meaning that we do not know which compounds are they capable to produce. Of course, if you will take the average genome of Bacillus, you will find plenty of biosynthetic gene clusters as well. But for some reason, there are no such variety of biosynthetic gene clusters and no such abundance of biosynthetic gene clusters in low GC gram positives if we compare to high GC gram positives, like actinomycetes. Why? This is a very good question, and if we knew, probably life would be much easier. But we can constatate that actinomycetes have large genomes enriched by biosynthetic gene clusters. Other soil gram positives also have biosynthetic gene clusters, but in much less amount than actinomycetes. Another thing is that actinomycetes have a very complicated life cycles, which have changing multicellular vegetative mycelia that develops into aerial mycelia that can form spores or even sporangia, similar to the one, for example, ascosporangia, asks. But this can be also somehow, this complicated life cycle would be somehow correlated with the size of the genome and with the abundance of biosynthetic gene clusters. But what are the evolutionary reasons of this? Where you have actinomycetes with large genomes, large cell cycles, and plenty of biosynthetic gene clusters. But you have other terabacteria which are not so complicated. It remains unclear to this point.
Let's thank Professor Yuchuk once more.
Okay.
Yes, thank you. So for today, we finished with lectures, and we will have dinner as usual at the usual place. So we'll go for the dinner, and tomorrow we will start at 9. And before you will have breakfast as usual. Do you have questions? Yes, and we will meet in this room tomorrow morning.
Okay. Okay, thank you very much.